Hi, my name is Ricardo de Sousa, and I'm going to present the study named Regular Physical Activity During a Gestation Exposed to Zika virus Inhibits Brain Atrophy, Behavioral, and Molecular Changes in the Offspring. Zika virus was firstly described in 1947 in Uganda, Africa. The main transmission vector is the mosquito Aedes aegypti. A congenital exposure to Zika virus can bring consequences to the central nervous system, such as microcephaly and also the development of Guillain-Barré syndrome, among others. Physical exercise is supposed to provide neuroprotection. Thus, the aim of our study was to investigate the neurological consequences of physical exercise during prenatal Zika virus exposure to mice pups. To do this, we performed the evaluation of the estrous cycle, as you can see here in B, and then we mated 2 female mice to 1 male mice for 24 hours. And then we evaluated the formation of a plug in the female mice to confirm the coupling. The swim training protocol started with a familiarization week that followed of 4 weeks of swimming training protocol that lasted 60 minutes during 5 days per week. The mating was done between the 5th and 7th day, and saline or Zika virus was injected in the embryonic day 10.5. As you can see in A, B, and C, there were no significant changes or difference between the body weight of the dams along the gestation or in the offspring at P1. However, there were significant differences between the brain weight of female and male pups at P1, where the Zika virus group presented a reduced brain weight, which was inhibited by the Zika virus swing group. Then we proceeded the evaluation of the anxious profile through the open field tests A and D, and the depressive-like behavior through the tail suspension tests B and E, and the forced swim test C and F. And what we saw was the presence of the anxious and depressive-like behavior in the Zika virus group in female and male mice, which were inhibited by the Zika virus swim group. Then we performed a Western blot analysis of the hippocampus of the pups. And then we decided to evaluate IBO1, that's a biomarker for microglia, GFAP, a biomarker for astrocytes, and BDNF, that's a biomarker for cell proliferation and survival. And what we saw was a reactive gliosis present in the Zika virus group, what was inhibited by the Zika virus swim group, and a reduction in the BDNF levels in the Zika virus group, which was inhibited by the Zika virus swim group in both female and male offspring. Congenital exposure to Zika virus has been described in scientific literature to provide change in behavior in mice pups. And physical exercise has been also described in scientific literature as capable of inhibited reactive gliosis and enhancing BDNF. Physical exercise in our study during a Zika virus infected pregnancy was capable to inhibit brain atrophy, activation of microglia and astrocytes, and contributed to the enhancement of BDNF, inhibiting changes in behavior. Thus, in conclusion, swimming for 4 weeks during prenatal Zika virus exposure prevents brain atrophy, changes in behavior, and disruption of molecular mechanisms in the hippocampus of mice pups. Thank you.